We are going to talk about monocot and dicot. And specifically, we are going to talk about the stem of monocot and root, I would say, root stem. We are going to talk on that behalf. So, before we start, we will like to learn about the two differentiator. What is monocot? What is dicot? I talked about them in the previous video. I'm not going to tell them again. Let's get on it. So first thing that we should find about in monocot and dicot is that inside the dicot is the superior one. It's the one with all the facilities that a kid could have. It has perfect vascular bundles. It has perfect system. It has perfect everything. Efficiency is the must in dicot. You can say that. But in monocot, it's not the same case. I mean, it has everything as well, but the quantity of the substances in the monocot is lesser than in the dicot. So when we talk about the dicot, what do we first find about? We find that inside the dicot that there are lots of things. First of any, if we took if we take a look of the sunflower stem, the TS of sunflower stem, if we take a look, the outer layer epiblema or endodermis, I think epidermis, not endodermis. It is the inside one. It is called endodermis. Epidermis is the above layer. It is a single cell layer made of parenchyma. Mostly the cells inside the tears of sunflower, you can say it is parenchyma. But when we take a look at it deeply, we find that the TS suggests that the cells are of single layer without any cuticle. Above the cells, they are present of trichrome as well. Now, underneath the cell, there's a cortex. Cortex is from the starting of the from the top of the, what do we call that, the top of the epidermis, and the bottom is the vascular bundle. In between that area, it is called as cortex. The cortex is divided into three regions. What are those three regions? The three regions are: first is happy or hemidermis. I think it's like that. It is something of H, I would say. And after that is general cortex, and underneath that is endodermis. It is hypodermis, I think. Hypodermis, general cortex, and endodermis. These have their own functions. Underneath that, before comes that pericycle, I think. Just leave it aside. Underneath the pericycle, there's slits. Inside the slits, there's pericycle. The function of pericycle: there are various type of cells, parenchymatous cells that are present there, and below that there are vascular bundles. What is vascular bundles? It is a thing. It is most important thing inside the plant, you can say. Why I am saying that? Because it does the conduction, storage of water, conduction of food, storage of food, everything. Transport system is controlled by these type of thing. So these are most important in my eyes. There are six, 2 to 6 present in monocot, I think. Yeah, 2 to 6 present in monocot, which is called as diarch and diarch or hexarch, and in dicot there are presence of more than 6 called as polyarch. So it's pretty interesting, I would say. In monocot it is polyarch, in dicot it is diarch and hexarch. Also, the cambium in dicot present. The cambium is the differentiating line between the xylem and phloem inside the vascular bundle. And what is the cambium? The function of the cambium is that it gives rise to secondary growth of the cells. It increases the girth of the body of the plant. So that's the function of cambium. So this portrays a really well structure of our monocot and dicot things. These are very essential for us to know, and you should know them as well. Thank you.